This is IAQradio, Indoor Air Quality Radio, the voice of the indoor air quality industry, with your host, Radio Joe Hughes, and the Z-Man, Cliff Zlotnick. And now, Radio Joe Hughes.
Good day and welcome to IAQ Radio Plus. We've got a great show today. We've got Dr. Karen Dannemiller from The Ohio State University. We're going to talk about understanding fungi, bacteria, and chemicals in the indoor environment. If you get a chance, check out our Facebook page at IAQ Radio or the YouTube page. Leave a comment, like it, or subscribe. You can also sign up for the weekly show announcement on our home site, IAQradio.com, where we've got all the shows archived and Cliff's blogs. Plenty of great information on there. Before we get started, let's thank our platinum sponsors.
IAQ Radio Platinum sponsor is John Don Products, where restoration and abatement contractors shop. Visit them at jondon.com. That's JONDON.com.
I also want to thank our gold sponsors, Particles Plus, Healthy Indoors Magazine, Gray Wolf Sensing Solutions, and AEML Inc. Laboratory. And of course, our association sponsors, the Indoor Air Quality Association and the Restoration Industry Association.
And now you can win a cool prize. It's time for the IAQ Radio Trivia Question. Be the first to correctly answer. Simply email your answer to czlotnick@cs.com. Or if listening live, just text your answer from your computer. And now, here's the Z-Man with this week's IAQ Radio Trivia Question.
Hello, everyone. Thanks for listening to IAQ Radio. I'm happy to report that Doug Conan, Aerotech Environmental in Dayton, Ohio, was first to identify September 21st, 1970, as the day that Monday Night Football debuted on ABC. The IAQ Radio Trivia Question for today, Friday, September 28, 2018, has been sponsored by Ideas, the solution chemistry company, creating unique solutions to odor removal, surface cleaning, and decontamination problems. Here is today's trivia question. Who insisted the addition of the word "the" would separate Ohio State from other colleges in Ohio? Back to you, Joe.
Whoever it was, they were correct. Anyway, this week we've got Dr. Karen Dannemiller. She's an assistant professor at The Ohio State University. Her interdisciplinary research integrates engineering with microbiology and addresses emerging health challenges and environmental concerns using an omics approach. We'll talk about that word a little more as we get into this. Dr. Dannemiller graduated with honors in chemical and biochemical engineering from Brown University, and she earned her PhD at Yale University in chemical and environmental engineering. During this time, she also completed an internship at the California Department of Public Health in their indoor air quality program, pretty widely recognized and respected program there. She also completed a microbiology of the built environment postdoctoral fellowship. Welcome to the show, Dr. Dannemiller. Do we have you on?
Great, thanks to be here, or nice to be here. Thank you.
I tell you what, you made me do my work this week. Reviewing those papers was a little challenging, but quite interesting, actually. Really enjoyed it. So what led to your interest in, indoor air quality, indoor environments, this type of research?
I was always interested in indoor exposures. So I started out in chemical engineering and was interested in exposures to formaldehyde in the indoor environment. And then when I started my PhD, I got interested in indoor microbiology and the exposures there.
Okay. And your PhD was up at Yale with Jordan Petya?
Correct, yes.
Okay. We had him on the show a while back now. Very interesting. Actually, he'll be the keynote at our Healthy Building Summit coming up October 25th. Looking forward to that and seeing him in person. So let's talk a little bit about your dissertation. Actually, I noticed when I was reviewing your CV, it was integrating measurements of environmental fungal communities with human health outcomes, which is, obviously like the big ticket, what we're looking at. How do we determine how environmental fungal communities and health outcomes are related, I guess. What do you think is the best way to actually measure environmental fungal communities when you're trying to figure out the potential health outcomes?
So like you said, I think this is the big question that's still out there. I've always been interested in this interdisciplinary field, basically bringing my engineering background to public health and looking at the indoor environment. So I think looking at these exposures and trying to figure out what to measure is a really important problem. It has a lot of health implications for all of us, and especially some vulnerable populations like kids with asthma. So I think the question of what to measure is the $1 million question, so to speak. And I think we're still trying to figure out what is the measurement that we can make that's the most associated with human health outcomes.
And we hear often that research using spore trap sampling and that using that to try and predict the potential for health effects just does not work. You mentioned a paper to me when we were talking earlier today. I'm sure you'll bring that up in the answer. Why do you think that doesn't work? Or maybe you can explain to listeners, what the findings have been when trying to correlate spore trap air samples using a slit impactor with health effects.
So the biggest thing that we want to consider when we're trying to determine what to measure is that we want the measurement with the best associations with health outcomes so that we can figure out what we need to measure to improve the health of the indoor environment. For spore traps, there's no consistent associations with these human health outcomes in the literature. And in fact, we're looking for the measurement that has the strongest association with these health outcomes. So there was a really great review written about this by Mark Mendel et al. In Environmental Health Perspectives in 2011. So I definitely point any interested listeners there as a really great resource. But that goes through looking at these different measurements. And in fact, the best measurements that they found that were the most associated with health outcomes were the subjective measurements. So things like visual inspection for mold growth or water damage, as well as moldy odor, have the highest and the most consistent associations with health outcomes. So in fact, that's probably what we need to be looking for if we're concerned about health in the indoor environment.
So again, a good visual inspection, olfactory is the key. But then, we also like to add, we get requests all the time. As an indoor environmental professional, I get, can you sample my air? I need mold samples. I need this, I need that. You do a lot of DNA-based analysis. How is that coming along with respect to the availability and economics for those of us out in the field trying to help people with these issues?
So I think at this point, we're still at a point where we're still trying to figure out the best thing to measure. And DNA sequencing is getting cheaper and cheaper and less expensive as we speak, and it's slowly becoming more accessible. So you have a couple different challenges in terms of accessing that in the field. The first is figuring out what to measure. The second is actually doing the measurement and getting that done. And the third is actually the analysis component, which at this point is still fairly challenging and maybe not accessible to typical practitioners, but we are getting there. So some of the reasons that quantitative measurements in the environment have not been associated as much with health outcomes, maybe things like there may be measurement errors in the exposure assessment, so we might be measuring non-causal factors. So with these analyses, we can now look at everything that's present in the environment, at least everything that's in our database, and try to find the best association. So try to find what are actually the causal factors in looking at these health outcomes. The second reason is that effects may change over time. Both the amount both the amount that's there may change over time and the effect based on age of the person that you're concerned about with the health outcome. And the third reason that it may be difficult to find associations with measurements and health outcomes is that you may have interaction effects from multiple exposures. So the benefit of these DNA-based techniques is that we now get a much more comprehensive picture of what is actually in the indoor environment. And we can start looking at things like those interactions, although statistically it's very complicated.
Well, and then we could even make it more complex if we add what we had in the title of the show, which was chemicals, and how moisture and microbial populations affect the chemicals that are in indoor environments. Any comment on that?
Yeah, I think there are absolutely interactions there. So some of the things that we've been working on in the lab, we see that there may be effects that chemicals have on microbes and effects that microbes have on chemicals in the indoor environment. So not only do you have that, but you're also going to have potential exposure effects as well.
Interesting. Very, very tough to tease it all out. Let's go to some of the papers here. The first one, John, is influence of housing characteristics on bacterial and fungal communities in homes of asthmatic children. This was in Indoor Air. I don't have the date in front of me here, but I'm sure you probably know. First, I want to ask you, in the title, you talk about housing characteristics. What do you mean by housing characteristics when you're in this particular paper?
So we defined housing characteristics fairly broadly for this paper. We were looking specifically for characteristics that had been identified in the past as potentially being associated with asthma outcomes. So what we decided on were number of people or children in the home, so number of occupants, the level of urbanization. So in this study, we were able to separate it out by homes in urban and suburban areas, single and multifamily housing units, so sort of the type of home, whether there was reported mold or reported water leaks in the home, the air conditioning use, as well as the presence of pets. So we defined it fairly broadly here.
I see. Okay, and now we've got that down. We've got the background. What are the key takeaways from this particular paper?
So the biggest takeaway from this paper was that we found that the building conditions within a home affected the microbial communities that people were exposed to. So we saw both fungal and bacterial communities were non-random, meaning that there were factors in the homes that influenced these communities. We also found that pretty much all of these housing factors that we looked at affect the microbial communities in some ways. So we saw associations with diversity or richness, and we also saw overall effects on the communities and different taxa that were present.
Now, we're going to talk more about diversity and richness. As it's kind of a theme throughout here, I think, at least in my review of things. When you talk about diversity, first, I guess, how many, you did DNA, next generation DNA sequencing on these. How many organisms are in the database that we're dealing with? I mean, it's just to give people an idea, just a ballpark idea.
There are thousands of organisms in the database, and we typically identify hundreds to thousands of organisms when we do these types of studies. So we get a really comprehensive picture of what's present in these homes.
I see. And just to give us a kind of an idea, what percentage of those organisms are bacteria versus fungi, and just a ballpark idea so we know in the database?
So, we use two different databases, one for bacteria, one for fungi. In terms of total concentration, they're actually usually about equivalent.
Oh, okay. Interesting. All right. Well, you also sent a paper or an article that was called Indoor Microbial Communities, Influence on Asthma Severity in Atopic and Non-Atopic Children. Let's go over the results here. I don't know if I can pull this up that way or not. John, can you go into yeah, there you go. Just pick it up a little bit, the background here. When I was looking through this, I saw among all children, increased asthma severity was significantly associated with an increased concentration of some allergenic fungal species, high total fungal concentrations, and high bacterial richness when using logistic regression in addition to microbial community composition by using the distance comparison test, which is probably a little above and beyond where we're going. I know you're using widely recognized methods for coming up with these results. So what is the summed allergenic fungal species? What does that mean?
Sure. So there are a couple of questions there. So the main goal of this study was to look at how microbial exposures in the indoor environment affected kids who already had asthma. So we wanted to look at their asthma severity in this study. And in this study, we separated out the children into two groups. So there were kids who were atopic, or those who were prone to developing allergy, and then there were kids who were non-atopic, so those who are not prone to develop to developing allergy. And then we sampled the dust and looked at all of the fungal and bacterial exposures that these children were exposed to and measured their asthma severity over the course of the following month. And when we looked at a couple different factors in the microbial communities that we thought might be associated with this asthma severity. And these included things like total fungal and bacterial concentration, overall diversity, a sum of allergenic fungal species, as well as the overall fungal and bacterial communities. And so for the sum of allergenic fungal species, we used a study that was done from 2008. The last name on it is Simon Knobe. It's in the International Archives of Allergy and Immunology. And from that study, they have a fairly comprehensive list of known fungal allergens. So we took that list of allergens and the fungal species associated with those allergens and summed it all together to get our measure of allergenic fungal species. And we found some pretty interesting things when we did this study. So we were able to comprehensively look at the microbial communities that these kids were exposed to and compare that to the asthma severity over the following month. And what we found is that the response of the children depended on their asthma subtype. So if you go and ask a physician how many subtypes of asthma there are, often you get a different answer. So I'll say that you can divide that a lot of different ways, but we broadly distributed them into allergic and non-allergic children. And what we found is that the allergic children tended to respond more to the types of fungi that they were exposed to. So things like summed allergenic fungal species, which makes a lot of sense. But it didn't actually matter how much they were exposed to. It was more sort of the question of who's there, which species are actually present. And then for the non-allergic children, we found that it was a total fungal concentration. So they responded more to how much they were exposed to. And this makes sense because based on their asthma status, they're going to respond differently to these environmental stimuli. The allergic children have more of an allergic type response, where the non-allergic children have more of an irritant type response.
Interesting. So they do have a response, but it's more of an irritant versus or asthma or increased asthma. Okay. Now, with that same paper, I highlighted the words significantly associated. Increased asthma severity was significantly associated with an increased concentration of the summed allergenic species, high total fungal concentration, high bacterial richness. When you say, I'm used to seeing associated with or cause, but that term significantly associated was new to me. What do you mean by significantly associated?
Sure, that's a great question. So we did a statistical analysis which showed that basically our p-value was less than 0.05. So we can say that our associations we found here were statistically significantly associated.
I see. Okay. All right. Next one is, well, I guess you kind of already told us what these means in layman's terms, right? I mean, you gave us a good, but if you want to maybe kind of give us a quick overview one more time of that, the results from that study.
From the study on the screen, fungal and bacterial growth in floor dust at elevated relative humidity levels?
Yeah, well, I was going back to the first one, the indoor microbial community's influence on asthma severity. I guess what I'm always trying to figure out is, for the people that are trying to either, clean their own home or figure out what's wrong with their own home, or those of us that are practitioners out there, what's the key takeaway for us from that particular paper?
I think there's two main key takeaways. One is that we did see associations with asthma severity based on the microbes that these children were exposed to. So their microbial exposure actually mattered in terms of determining their asthma severity. And then the second takeaway is really that basically that it's complicated is the large takeaway, that their reaction depended on their asthma subtype. So for the kids with allergic asthma, it mattered more what they were exposed to. And for the kids with non-allergic asthma, it mattered more how much they were exposed to. So I think overall, I think that the picture is still pretty complicated. We're still at a point where we're looking to make recommendations, and we're maybe not quite there yet. In terms of asthma severity, I feel like a lot of the standard recommendations are still good advice. Things like reducing overall exposure to dust is potentially helpful, because it might help both of these kids, especially if you can reduce exposure to allergenic species, but also if you can reduce exposure to the total fungal concentration.
All right, there was another thing that caught my eye in that paper. The fungal genus Volatella, I may be pronouncing that wrong, was associated with increased asthma severity in the atopic children. This goes back to what you were saying about the kids that were atopic for allergy. They reacted more to specific allergenic fungi. And this one is kind of new to me. Can you talk a little bit about that?
Absolutely. So we were also surprised to see this association with Volutella pop up. It's typically a turf grass pathogen, so it's not something that we really had on our radar before doing this study. So I wouldn't necessarily at this point recommend that everybody go out and look for Volutella in the environment, because we're still not sure if this is a causal association. We probably want to try to repeat this result in other studies. It's still possible that it's associated with something else that's associated with asthma. So at this point, this study is more flagging Volutella for further research. So I wouldn't go out and look for it quite yet.
Okay. All right, good. That's good to know. You also note here that the yeast genera as Condoa might be protective, and Cryptococcus species might also affect asthma severity. That's interesting.
Correct. So we're looking at these particular species now because they've shown up in a couple different studies at this point. So we also did a study looking at asthma development and found potential associations with species in the genus Condoa, as well as diversity among all the species within the genus Cryptococcus. So I'll talk about Cryptococcus briefly. So this is a genus within the phylum Basidiomycota, and what that basically means is that it's generally been understudied because a lot of these are difficult to culture. So in the past, when people were more reliant on culture-based techniques, it was a lot more difficult to study. And now that we have new DNA-based methods, it's a lot easier to study species within this genus. And so we found associations with Cryptococcus, especially in our asthma development study. And when you look to the literature, you see other associations with Cryptococcus. So one of the species in this genus, Cryptococcus neoformans, is also a known human pathogen. Absolutely. And there's been studies that have been done, both epidemiological studies and as well as a rat model, that found that subclinical infections with Cryptococcus neoformans are associated with asthma. So these subclinical infections with Cryptococcus are typically low-level persistent infections that some children might develop. In the doctor's office, they're often confused for viral infections because they don't respond to antibiotics, but they're actually fungal. And so children who have developed these infections may be more likely to develop asthma. So we thought it was interesting that we find associations within the same genus in our studies as well.
You know, with the Cryptococcus, were you finding a lot of pigeons or bats or birds, infestations and nests in these houses? Because typically that's a common place to find Cryptococcus.
Absolutely. So Cryptococcus is present in a lot of different environments, and animals are one source of it. They're known for being present in pigeons, like you mentioned, especially. We didn't see anything like that show up in our data specifically, but we also didn't necessarily look for it. It wasn't something that we were looking for when we went into this study. So it's an open question about what the specific sources of Cryptococcus species were in these studies.
Thanks.
Interesting. I kind of suspect that in the past, we've not found Cryptococcus as often as you are now because, like you said, the culture and other techniques we use just don't pick it up as easily. Does that seem like it makes sense to you?
Absolutely. I think it's a lot more difficult to detect using culture-based methods or other techniques. So the DNA-based methods are really helpful in terms of looking at it.
When you do all this research you're doing on indoor environments using the DNA-based methods, do you commonly find Cryptococcus species in the indoor environment?
Yes, so there are about 304 different species of Cryptococcus that we know about, and we generally find maybe up to about 30 of them commonly in our studies of the indoor environment. But it is a very common genus. We find many different species present in our samples.
Very interesting. Very interesting. All right, let's go to the next one. This is Volatella. We talked about the Volatella, I believe. And let's go to the next paper here before we get to halftime. Let's go, unless you had anything else you wanted to say about that previous paper, the article, I guess it was, Indoor Microbial Communities' Influence on Asthma Severity. What's the key takeaways on that one, I guess?
On influence on asthma severity. Yeah, so I think the key takeaway is that we did find associations between these microbial exposures and human health outcomes. These were not homes that were necessarily selected based on things like water damage or mold growth that you could see, but we did find general associations with these health outcomes. So I think it's something that we need to keep looking at into the future. We know that there are health outcomes there, and try to learn more about both what we can do to improve the indoor microbiome and prevent these health outcomes, and how that will potentially be beneficial for children.
I'm glad you brought that up because I didn't ask that in the very beginning, and I think that's an important point to bring up. That how were these houses chosen now?
So these houses were chosen actually as part of a broader study that had already been completed, part of the STAR study that was done in Connecticut and Massachusetts. And in that study, they looked at a range of homes, so urban versus suburban homes. And so they had these dust samples left over. So we selected these dust samples for our analysis based on the atopic status of the child. So we wanted about half atopic, half non-atopic for our analysis.
I see. It had nothing to do with the conditions of the home.
Correct, yes.
All right. Very good. Very good. Let's go to, what time? 12:26. We have time to get into number 3 here. This one is called Fungal and Bacterial Growth in Floor Dust at Elevated Relative Humidity Levels. And this is the one that actually caught my attention when I started checking around and figure out, I know we had met before, but we hadn't had a chance to connect and get you on the show. And this one, I was like, all right, I've got to track her down and get her on this show here because I just found this fascinating, especially because I find a lot, especially now in my practice, elevated relative humidity is a huge issue that I think is undercovered. In other words, people don't talk enough about the issues with elevated relative humidity and how common it is, especially in the swing seasons. I think people know that during the summer when it's hot and humid, you get elevated relative humidity. But in my experience, the biggest problems with elevated relative humidity come in those swing seasons in the spring and the fall. And this year, it's been all year. So this one I found fascinating. Let's talk a little bit about it. First, what led to your interest in this floor dust and elevated relative humidity, the topic?
Absolutely. So some of the same reasons that you just mentioned. So we were interested in looking at the effect of elevated relative humidity on microbial growth in floor dust. When people think of the indoor environment, a normal indoor environment that's not that does not have any known water damage or moisture problems, we typically think that the microbial communities there are pretty dormant, that they're tracked in on our shoes from soil outside or deposited from indoor air, especially the fungal communities, maybe the bacterial communities deposited by occupants, but they don't necessarily think of it as an active community that experiences growth. So we wanted to ask the question, is elevated relative humidity sufficient to support microbial growth in the indoor environment? We selected carpet for our study because carpet has fairly high resuspension rates compared to a solid surface flooring. So we wanted to look at the dust in the carpet and see if elevated relative humidity would support this growth. So we collected carpet from a home in eastern Massachusetts, and we took it back to our lab. We embedded dust from the same home in the carpet, and we subjected it to relative humidity conditions of equilibrium relative humidity conditions of 50%, 80%, 85%, 90%, 95%, and 100% equilibrium relative humidity. And then we measured the microbial communities in the dust before that incubation, as well as the dust in the communities after that incubation. And what we found is that indeed we did see microbial growth in the dust under sufficient moisture conditions. So we're not talking about a situation where we have condensed water. We're talking about just elevated relative humidity conditions. What we saw is we didn't see an increase in numbers at 50% relative humidity, but we saw an increase in fungi starting at about 80% relative humidity. And that increased all the way up to about 100% relative humidity, where we saw more than 27 times increase in the fungal concentration in the dust from carpet samples that were incubated at that relative humidity levels compared to in the original dust. For the bacteria in this sample, we did not see an actual increase in numbers at the relative humidity levels, except at 100% relative humidity, where we did see an increase in total bacterial concentrations. So the main point of the study was that elevated relative humidity was sufficient to support the microbial growth in these carpet samples. You didn't necessarily need condensed water to be present.
Don't bacteria need a lot more water than fungi, and that's kind of why you found what you found?
Absolutely. So the fungi are much more resistant to desiccation, and so they're more able to use water that's available at lower equilibrium relative humidity conditions than bacteria. So we had only seen the bacteria increase in number at 100% relative humidity. Right. We did see a change in community structure for the bacteria that were incubated at 95% relative humidity, but we didn't necessarily see an increase in number. We just saw an increase in basically who's there. We also continued these incubations out for about 6 weeks. So our initial analysis was done after a 1-week incubation at these different relative humidity conditions, but we continued it out for about 6 weeks. For the fungi, we ended the experiment after about 3 weeks because the carpet squares were essentially covered in visible fungal growth. So we said, hey, I think we proved our point here. But at 85%, we continued out to about 6 weeks, and we saw a continued growth as well over that period of time.
Interesting. When we come back, we have to stop and thank our sponsors, but when we come back, I've got a handful of questions I'd like to get into a little more detail on that study. We'll be back for the second half of our interview. We've got Dr. Karen Dannemiller from The Ohio State University.
IAQradio Platinum sponsor is John Donne Products, where restoration and abatement contractors shop. Visit them at jondon.com. That's JONDON.com. Gold sponsors are Particles Plus Engineers and Manufacturers of feature-rich particle counters and air quality monitoring instrumentation. Learn more at particlesplus.com. Count on us. Healthy Indoors Magazine, a free online digital magazine for industry professionals and consumers. Subscriptions available at healthyindoors.com. And AEML Laboratories, free FedEx shipping, great pricing, same-day results, and never a rush fee. Learn more at AEMLinc.com. Gray Wolf Sensing Solutions, who use advanced sensor software technology and embedded computers to provide superior environmental test instrumentation. Visit them at wolfsense.com. Association sponsors are the Indoor Air Quality Association, a multidisciplinary organization dedicated to promoting the exchange of indoor environmental information through education and research. Learn more at IAQA.org. And RIA, the Restoration Industry Association, the granddaddy of the restoration industry. Network with leaders. Learn more at restorationindustry.org.
Okay, we're back for the second half. We've got a couple text questions. I just want listeners to know I will get to those. And in fact, I think you've anticipated some of my questions. First, this is your Ohio State Indoor Environmental Quality Research Team. We wanted to make sure we got those, the gang up on the video here. So you want to mention anything about your group here?
Yeah, absolutely. I couldn't do any of this without the students who are in my group. So those right now, our group consists of Ashley Bope, Sarah Haynes, Samuel Cochran, David Kormos, Nick Natashi, Iona Campbell, Victor Emma Sodor, and Nicole Renninger. So definitely, I'm really thankful to work with some really fantastic students and participate in the work that they, in the end, they do a lot of the work and most of the work. So it's really great to work with them.
It's a good sized team you got there. All right, let's talk a little bit more about the methodology here, how you did this. First, how did you measure equilibrium relative humidity?
So the nice thing is for this study, we were working in a laboratory, so it's a lot easier to do in a laboratory than it is in the actual environment. So we used these glass chambers that were sealed with parafilm, and we controlled the relative humidity in these chambers using a salt solution. So we made a salt solution at a specific water activity, which could then control the relative humidity within the chamber. And we measured the water activity of the salt solution, and then we also measured the relative humidity in the chamber using some monitors for temperature and relative humidity to make sure everything stayed where we needed it to be. And then these glass jars sat in a larger incubation that controlled the temperature at a constant level.
Okay, and you chose 50%, 80%, 85, 90, 95, and 100. And as I'm sure many listeners know, EPA recommends 30 to 50%, never above 60. ASHRAE and others try and keep you below 60. And I've got a text question. She'd be interested in your thoughts on the impact at 60, 65, and 70% relative humidity, which are more common than what you're showing here, 80, 85, 90.
Absolutely. So I think that's a really great question. We selected these relative humidity conditions because we weren't sure what we'd find going into this study in terms of the growth at these different levels. So we selected 80% as a set point because that's been shown in other systems to support microbial growth. So on different building materials like wood or drywall or things like that. I think so 80% is particularly high for a home. Hopefully you don't see that on a regular basis, at least in homes that don't have problems. So these might be fairly high results, but you could see levels this high in specific situations, such as in a bathroom during a shower where you might have elevated relative humidity conditions, or in certain climates if you're working in a home with no air conditioning or something like that. But I think there are more realistic relative humidity conditions that could still potentially show growth, and we actually have some samples running now to look at potential long-term growth. So we may just need to be more patient at the levels closer to like 65-70% relative humidity to see that growth down the road. The other thing that we're working on is to make these studies more realistic to building conditions. So right now my student Sarah Haynes is working on a study to look at fungal growth in these samples at varying relative humidity levels over the course of a day. So in a typical home, you might have relative humidity that increases and decreases. And so we're exposing some of these samples to elevated relative humidity conditions for shorter periods of time. And so we still see fungal growth for the samples that are exposed to elevated relative humidity levels, even if it is over a shorter period of time per day.
That's interesting because there's been thoughts in the practitioner world and in the training world that it takes longer than I think people have shown in the research world. It takes longer for mold to grow on building products, and that 24 to 48 hours is probably extra cautious, I guess you would say. But have you seen growth that quickly in your laboratory?
So I think we haven't looked at such short timescales as 24 to 48 hours. We definitely see the dust uptakes the moisture out of the air very quickly. So after about a day, you can see that the relative humidity, the water activity of the dust has equilibrated fairly well with the water activity level. It's not, maybe doesn't quite reach it at equilibrium yet, but it definitely uptakes the water very quickly. And I also predominantly study fungi, which grows a lot more slowly than bacteria. So the bacteria is going to grow a lot more quickly than the fungi necessarily.
Interesting. All right. Tell us a little bit about what you did find at 50% equilibrium relative humidity. Was there any change?
In our study, after 1 week, we did not see any change in total numbers of increase in spores in this study at 50%.
And I think I noticed I was looking at some of the other papers you used, and I think you chose 50%, correct me if I'm wrong, because there's a good bit of data out there on dust mites and dust mite activity at 50%. Is that somewhat accurate?
We were looking at that, but we were also considering the EPA recommended levels, and we knew that the EPA recommended 50% as the ideal upper limit for relative humidity. So we wanted something that was close to our elevated conditions, but still within the acceptable range. And that's why we chose 50% predominantly.
Okay. All right. So let's move up a little bit. At 80%, what did you start to see? You know, what were the changes that I guess we could kind of summarize? You did already a little bit, but as far as, I don't think you went into the richness and the composition. Can you talk a little bit more about that?
Absolutely. So in terms of composition, we saw the types of fungi in particular show up that you would expect. So the typical species that you might find in a damp building. So things like Aspergillus and Penicillium and Willevia were the predominant genre that we found starting to show up at 80%, and especially at the higher levels at 100%. So typically at the slightly lower but still elevated relative humidity levels, we see Willevia show up predominantly, so maybe around 85%. And Willevia will continue to increase in total numbers of spores up to 100%. But then above that, we also see things like the Aspergillus and the Penicillium showing up that tend to grow at the higher relative humidity levels.
Yeah, so it seems like Willevia, which I don't think we focus enough on either, that grew at a different range?
It did. So it continued to grow at higher levels at 100% compared to 85%, but it showed up a little earlier in terms of relative humidity levels. And the other thing that you asked about was also the overall diversity and richness. And we see a really complicated story with diversity. I'm referring specifically to richness, which is the specific number of species that we find. So when we're measuring so many species, we can look at how many different species we actually find to be present. And there's a fairly complicated story there, both in terms of the health outcomes and where we see richness increase and decrease in a building. For fungi, it seems to be that if you measure richness in a building that is affected by water damage, but if you take a measurement away from the water damage, you see an overall increase in diversity. But if you take a measurement right where the water damage is, or in an area where you see that actual fungal growth, you tend to see a decrease in diversity. And that's what we saw here at our elevated relative humidity levels. We were sampling right where the water was, and so we saw a decrease in overall diversity with increased relative humidity levels. But I'd like to note that richness is a fairly non-specific term. So we're looking at just the total number of species present. We don't necessarily know what they are. So there's this complicated picture with richness, and we're still trying to figure out exactly what's going on.
Interesting. All right. Now, I also noted when I was reviewing that you looked at resuspension and came to the interesting conclusion that our material balance model predicts that at growth due to 100% relative humidity for 1 week, the resuspension of microbes grown in carpet dust may account for 75% of the fungal and 21% of the bacterial load in the air. Your main point appears to be that we underestimate the role of resuspension of microbial contamination when it comes to carpet, at least in this study. Is that accurate? And is there anything you'd like to add?
So we wanted to see if this additional growth could potentially impact human exposure. And this is under very specific conditions that probably wouldn't normally occur in a building. So hopefully nobody is intentionally leaving their house at 100% relative humidity for a week. I think you might have problems if you did that. But for our chamber, it was exactly at these relative humidity conditions for 1 week. So we wanted to estimate what that potential contribution from the growth was. And our overall finding was that yes, the growth could indeed increase the amount of microbial exposure that you're exposed to. And this was based on a rough resuspension model, so assuming that this growth occurred in carpet and then you walk across the carpet, you're resuspending it into the air. So that was our main conclusion, that the growth could potentially impact human exposure.
And it sounds like this has led to other research that you're doing now. Can you kind of elaborate on that a little bit?
Yeah, absolutely. So the one project that we're working on now is to try to make this look at this under more realistic building conditions. So under conditions where relative humidity varies throughout the day. And then we've also looked at gene expression and microbial function in these dust samples. And so we did a meta-transcriptomic study. That's a really long word that basically means we looked at all the different genes that express all the microbial function that's occurring in these samples and asked, what are they doing? So we know they're growing, we know they're active, and we wanted to know what they're doing.
Gotcha. And that's the next paper we want to talk about here, is the gene expression of indoor fungal communities under damp building conditions, implications for health. But before we do, we want to go to our roundup here and make a clip. I want to make sure I give you time to ask any questions you have. But we'll be right back.
I'm still trying to catch up writing the blogs.
I'll bet you are.
It's the 2018 Healthy Building Summit, October 25th through the 27th at Seven Springs Mountain Resort in the gorgeous Laurel Highlands of southwestern Pennsylvania. Network and practitioners, prominent researchers, and industry leaders in an intimate and informal setting. This year's theme is IAQ, Remediation and Restoration, Research to Practice. This is the only industry event that performs live research and links researchers and practitioners. Marquee sponsors include Hayward Score, helping people live healthier lives, and Particles Plus. Count on us.
All right, we're back. Let's get into the roundup. Cliff, before I jump in here, do you have anything you wanted to ask about the first three papers or any of the first part of the interview?
I do not.
All right, now we're into paper number 4. John has it up here. Gene expression of indoor fungal communities under damp building conditions, implications for human health. All right, so gene expression is kind of a new term for us here, but I think you mentioned what it was briefly prior to going into this, but maybe you give listeners just a little more on what you mean by gene expression.
Sure. So we had looked at these carpet samples in the previous study, and we knew that there was microbial growth occurring in the carpet samples. And so the big question we had left is, what are they doing? What is the microbial function? What does the microbial function actually look like? And to do that, we wanted to study gene expression. So we actually looked at the RNA that these microbes produce. So microbes have DNA, and then they create RNA when they express genes. And so we measured the RNA to get an idea of what these microbes were actually doing in these dust samples under elevated relative humidity conditions.
Okay, and then I guess this goes back to the introduction, using an omics approach. I don't know if I got that right or not. Did I pronounce that right? An omics approach you're talking about here. And then we're talking about a meta-transcriptomic study. And you mentioned that before the break, but if you wouldn't mind, I wonder if you could kind of break that down in layman's terms for us here.
Sure. The meta-transcriptomic study is basically a long word that means that we looked at all the different genes that were expressed in these samples. So oftentimes people might do a transcriptomic study, and that's looking at maybe a specific species that you're interested in to look at the transcriptome, basically all the genes that are expressed, the function of that. We looked at the meta-transcriptome, which means that we looked at the entire community in these samples. So we wanted to know what all the different species were doing together in terms of microbial function.
And when this occurs, the gene expression, is that when we start to see things like the secondary metabolism of these organisms start to kick in? Am I confusing that?
Yeah, so we had some really surprising findings from this study. The first surprise, for this study specifically, we looked at 50%, 85%, and 100% equilibrium relative humidity. And the first surprise was that at 50%, we actually saw expression of genes. So we had originally intended that as a control sample. We didn't actually think we could get RNA or see gene expression occurring at 50%. But indeed we did. And we saw that these microbes were active, but they were really participating in primary metabolic processes. So they were really just hanging on, kind of waiting for some water to come by so that they could grow. So they weren't doing too much, but lipid metabolism was complete, carbohydrate metabolism was complete, and they were still having some microbial function. And then when you looked at 85% and 100% equilibrium relative humidity, we saw increases in gene expression, especially at 100% equilibrium relative humidity. And when we looked at this, we saw that at 85%, the microbes were still participating in primary metabolic processes, so basically just kind of hanging on, trying to remain there what they're doing. But at 100% equilibrium relative humidity, we saw a lot of secondary metabolic processes. So they started to expand out what they were doing. And they expanded out in pretty diverse ways. In fact, our different samples at 100% equilibrium relative humidity were actually very different even from each other, as well as being different from the 85% and the 50% equilibrium relative humidity. And that's particularly important because a lot of things that you might be concerned about in the indoor environment, so allergens, mycotoxins, things like that, often tend to be in those secondary metabolic pathways. So we actually saw an increase in the number of allergens and mycotoxins produced in the samples that were incubated at 100% equilibrium relative humidity levels. So this brought up a really interesting question for us. So when you measure a spore, the question is really, is a spore, or are they different depending on how they've actually grown? So you might have a very different response from a health standpoint for a spore that you're exposed to that's been grown at these higher relative humidity levels.
Interesting. John, can you pull up that last slide, the one that had that changed? I want to ask Karen if she'll explain this one to us, because it has to do with this gene expression, right?
Yes. So on this slide here, you can see a map. So this looks very complicated, but this map shows all the different genes that we found were expressed in our samples. And if you look at the top left corner, you can see the water activity levels that we were looking at. So first at the 50% equilibrium relative humidity levels, we just see those primary metabolic processes that are lit up. At 85%, we see a little bit more, but it's still largely those primary processes of what the cells are doing. And then at 100% equilibrium relative humidity, we see all the secondary metabolic processes that you see here that light up. And so we see a lot more gene expression as well as very diverse gene expression. So we see sort of an increase in what we think of as metabolic diversity at these higher equilibrium relative humidity levels.
Very interesting. Okay. Let me see. Cliff, anything you wanted to...
The one thing, Joe, I just wanted to pick up and get an answer for that you had mentioned was omics. You promised you were going to tell us what omics were.
Yes.
Yeah, omics is looking at basically a lot of different factors, a lot of different biological molecules, and looking at how that whole picture relates to things like microbial function or structure of microbial communities, things like that.
Okay, thanks.
And I'm wondering, let's go back to that graphic, John. I think that's fascinating. Can you tell us a little more about what we're looking at? I mean, I know we're looking at the gene expression, but how did you build this model? Is that a computer program, and then you kind of sliced and diced in the sample results there somehow? How does that work?
Sure. So a lot of the bioinformatic analysis here was done by Bridget Haggerty, who's a PhD student at Yale University, working with Jordan Peccia. And so she did a really fantastic job working with this data. The data is really complicated. So first, it's extremely difficult to actually extract the RNA from the dust itself because of the presence of RNases. So you have to be very careful, use a very specific technique to get the RNA out. Once you have the RNA out, you sequence it, and then you basically have this very large file. It can be gigabytes of data that you then need to process from the RNA reads into what's actually going on here. So she used primarily a program called Trinity to basically take those raw reads and turn it into what you see here, which is a map of genes that are expressed. And if you look more closely at this figure, you can see different pathways. So you can see like carbohydrate metabolism is labeled, lipid metabolism is labeled, and you can see that those pathways are complete and they're being expressed at all three different equilibrium relative humidity levels that we measured.
So the foundational one, the primary ones at all three were expressed, but then as you went up in relative humidity, you had more connections essentially between them. That's fascinating. Very interesting. Energy metabolism, amino acid metabolism, carbohydrate, lipid. It gets more confusing every time I talk to you researchers. Although you're helping me understand it here very, very much. Now, let's go back again to the results of the study. I mean, how are the results of the study going to affect the study of health effects and damp indoor spaces? I know you mentioned that a little briefly a moment ago, but I want you to kind of recap that for the listeners, please.
So I think based on this work, we're still trying to answer the question that we started with at the beginning, which is what is the most important thing that we need to measure in the indoor environment to find associations with human health outcomes. So what we found in this particular study is that we may need to be looking at something different because we see different biological molecules that we know may be associated with health outcomes like allergens and mycotoxins. And we see them expressed differently depending on how the microbial communities were grown at different equilibrium relative humidity conditions. So I think basically the conclusion right now is that looking at moisture in the indoor environment is probably the most important factor. If we can control the moisture, we can likely create a healthier environment, and that's been shown with associations in the literature between dampness and the environment as with health outcomes. So I think the end result right now is we still need to keep looking at the moisture, those visual signs, so mold growth, visible water damage, things like that, as well as moldy odor, and that seems to be the thing that has the strongest association with health outcomes at the moment. And in the future, as we continue to do these projects, we may get a better idea of other specific things that we can quantitatively measure that may be more strongly associated with health outcomes. But I'm sort of waiting until we find something that has a stronger association than these other more subjective factors before we switch over to making those measurements in the field.
So let me make sure I got you right. It seems at this point that the odor, the microbial volatile organic compounds are most clearly related to health issues, or that's where you most commonly find health issues?
Yeah, the more subjective inspection, so visual evidence, so visual mold growth, visual water damage, things like moldy odor have the stronger associations with health outcomes at the moment. And the reason for that may be because of what we're finding here. So we see that these microbial communities at these elevated relative humidity conditions produced more allergens. So even if you're measuring the same number of spores, if they experience different water activity levels, you may be exposed to a different number of allergens. But I think that in the end, I'm a researcher, so I'm always going to say more study is needed. And I think that's where we're going with this.
I think that's fair enough. So it seems like, as we've always pretty much known, that at the higher relative humidities, you have more secondary activity and people seem to have more health issues.
Yeah, and absolutely.
I also wanted to point out that earlier, and it might have been the third study we talked about where we were looking at the different relative humidities, you pointed out there, and I think it's an important tip for those of us doing inspection work. Somewhere in the paper talked about slab and slab on grade with carpet right on top of slab on grade. Could you just kind of comment on that for a moment?
Yeah, absolutely. So I think we need to keep in mind the different relative humidity conditions over the entire space of a room. So you may have different relative humidity levels at different points. If you measure the relative humidity in the center of the room, it could be very different that towards the edge of the room or in the carpet. So there have actually been studies that have been shown that the relative humidity in the carpet can be very different than in the center of the room. Or you think about other areas where you might potentially see things like condensation happening. You might have, even if you're not seeing condensation, you might have different equilibrium relative humidity levels at different points.
And high enough that you're getting this type of gene expression and activity going to your last paper that may lead to more allergenicity and toxicity and all the other things we worry about.
Yeah, potentially. So I think we really need to look at the moisture in the indoor environment and make sure that we're maintaining it at levels that we find acceptable.
Karen, I really appreciate you, Dr. Dannemiller, joining us today. I just want to add, and Cliff, do you have anything you wanted to add?
I do not.
You got your work cut out for you today with the blog. I know that. Anyway, before we go, we always like to give you the last word. Is there anything we missed? Anything you'd like to add? Any final thoughts for our listeners?
I'd just like to thank again all the people that have worked on these various studies with me. So I'd also like to thank the Alfred P. Sloan Foundation, who funded a lot of this work, and the various other funding agencies associated with the papers, all the co-authors, as well as the students in my group that I mentioned, as well as the participants of the studies, without who this wouldn't be possible. And in the end, I think it's really great to be having these conversations and talking about moisture in the indoor environment. I'd love to hear from any industry professionals out there who are interested in what they heard today and hear what they think about it. But in the end, I think we really need to be paying attention to moisture in the indoor environment as an important factor when we're concerned about health outcomes in the indoor environment.
Well, thank you so much. We really appreciate it. And again, I look forward to getting you back in the future and following your research as time goes on. I think it's going to be fascinating to continue to look at these issues. But I want to thank you, Dr. Karen Dannemiller, for joining us from The Ohio State University. I also want to thank my co-host, the Z-Man, Cliff Zlotnick. Going to have a busy afternoon writing his blog. At the controls, John, you got to have faith. Most importantly, our growing group of loyal listeners, check us out on YouTube or on Podbean. If you're just doing the podcast, you can download those and play them in your car. We'll be back next Friday at noon for the next episode of IAQ Radio.
For IAQ Radio, I'm Spike Reed saying thanks for listening.